Well, I'd like to start by thanking everybody for attending this exciting virtual ISMB meeting. And what I'm going to talk about today is patterns of variation in protein sequence evolution. This is a topic that's excited me and my students for some time. And if we think about variation in patterns of protein sequence evolution, there are a number of dimensions we could approach this question from. We could think about patterns of variation across sites within proteins, variation among proteins, or variation among groups of organisms. We're really going to focus on the last two. And we'll use a relatively straightforward method. We'll start with estimating clade-specific models. So we'll take large training data sets of arbitrarily chosen proteins, in this case from 6 eukaryotic clades, 3 vertebrate clades, as well as plants, oomycetes, and yeast. For comparison, we'll have another set trained on broadly sampled proteins across all of the above, as well as some other additional microbial eukaryotes. Then we'll estimate exchangeability parameters for each pair of amino acids and ask some straightforward questions. Do the models differ, and are the models predictive? Well, do the models differ among clades is really captured on the vertical axis of this sort of hypothetical parameter space. Down in the lower left, we see very little difference among clades. Pretty much all of the proteins are clustered on top of each other. And as we increase variation among clades, we see groups of organisms, here defined by colors, separating out. The other axis we can think about is variation among proteins. These are the individual colored dots. And of course, in the upper right, we have high variance among clades, high variance among proteins. Well, to assess variance among clades, we can simply look at the distances among models. If we cluster models by the distances among those models, here's a midpoint rooted clustering diagram, we see 2 clusters. One comprising the vertebrate models, as well as previously published models for viruses that infect vertebrates, and then a second cluster that comprises all of the non-vertebrate models, as well as a wide variety of previously published models. Those previously published models were all trained on diverse proteins sampled from many different groups of organisms. Now, if we want to get a handle on variation among proteins within each of these organisms, one way we can do this is by cross-validation. What we're doing here is we take a validation set of proteins that are not included in the training data, and then we ask whether we can use the models as a classifier for proteins from this cross-validation set. And what we find is, yes, we can. 70 to almost 90% of the time, we can identify the group of organisms, at least to the level of vertebrates, plants, yeasts, or oomycetes. Within vertebrates, there's a little bit dicier in terms of the identification. The eukaryotic data set is a little more spread out over existing models. Some of this is because of taxon sampling. Since the patterns of protein sequence evolution are varying among taxa, we have some sensitivity to the taxon sample in a broadly sampled data set. However, what I feel pretty comfortable saying is we're probably in this part of parameter space here where I've circled, where there's a fairly big differentiation among groups of organisms and reasonably tightly clustered near sort of a best model for each clade. There certainly is variation among proteins within each of those clades, and certainly, although we didn't look at it here, variation within proteins among sites. What are the differences among models? Well, they can largely be explained by weaker purifying selection in vertebrates, no doubt due to the smaller effective population size. Specifically, those exchangeabilities that are elevated in vertebrate models typically involve larger changes in amino acid side chain volume. So you get radical differences in amino acid size. Let's move beyond these eukaryotic models to the whole tree of life. And I'm going to tell you about examination both among bacteria, archaea, and eukaryotes, and differentiation between sort of the broad ensemble of proteins and ribosomal proteins. We can use the same methodology, although here, as I said, I'm going to use many different groups of organisms, as well as breaking it down by ribosomal protein and all protein. So, do the models differ? Well, yes. We see a clear differentiation between bacteria on the one hand in blue and an archaeal eukaryotic group in green and red. Within each group, there's no real congruence with the species tree. This is ribosomal protein models. What if we go to all proteins? Something very similar, a little more complicated because we have the methanogenic archaea coming out with one specific group of bacteria. However, they are united by a very short branch. So a very similar pattern. We see something similar if instead of using clustering, we use non-metric multidimensional scaling. We can differentiate between groups of models. Let's put everything together, ribosomal models and the all-protein models, and you see clear differentiation. The gray is the general models based on a large ensemble of proteins, and slightly more spread out are the ribosomal protein models. But we do differentiate between bacteria on the one hand, archaea and eukaryotes on the other. So, there is phylogenetic structure. Model has changed over time. The predictive ability of these models varies a lot more than in the previous slides. For example, halobacteria are quite predictable. Within bacteria, we can usually identify them to the level of bacteria, but not to specific groups very well. Within the groups, there is no congruence with the species tree. So this could be consistent with a single shift in model parameters. With that, I'd like to end and see if there are any questions. Thank you.